But especially with these drop-based or any limited dilution methods like these microwells, is that you always get doublets. So in some experiments like mixing human and mouse, then it's fairly easy to distinguish them, right? Because if you have reads them up to both genomes, you know they're doublets. But of course, you will have some doublets of human to human and mouse to mouse that you can't distinguish. Or in this case, mixing two cell types, and you get some cells here in the middle that express genes from both cell types. There are some estimates on the 10x website, some tables on like the estimated rates of doublets depending on how many cells you loaded and recovered, where you can know in advance that, okay, I know that 5% of my cells are probably doublets, and keep that in mind all the time when you do analysis. The problem is knowing where are my doublets. Another thing is actually that cell debris can also cause doublet signatures. So this is from a project where they did single cell RNA-seq on frozen samples. And for some reason, these two samples, they thawed them and then the machine was not working and then they froze them again. And then when I ran doublet prediction on these samples, I got a really high doublet prediction for these refrozen samples. I don't know if the cells stuck together or broke or something, but the quality of the data can also influence how many doublets you get. So it's not a perfect linear relationship like they present on the 10x website. And it might be different for different tissues and so on, and dissociation protocols, how many doublets you end up with. As Carolina mentioned, cell hashing can help you to see doublets, but only doublets then that occur between two samples. Because since we add these antibody barcoded or antibodies with barcode to two samples and then mix them in the library prep, we can see when we have one droplet with two barcodes, then we know that we have two cells in there. And a lot of these doublet detection methods have been trained on this kind of data that we know are doublets. But in most cases, you will probably not have hashing on your data. And of course, you might have doublets of cells from the same sample. And if they cluster together with the doublets with two hashing barcodes, then you can probably say they are also doublets. But other doublets of a cell type that's unique to a sample, you can't say anything about. What you often see once you do UMAP or t-SNE, if you sort of have these streaks in between clusters, it might on one hand, of course, be like a differentiation trajectory, that you have one cell type going to another cell type, but it might also just be doublets. So you have to be really careful with what you believe to be doublets. And also when you do trajectory analysis, not to make any sort of assumptions about differentiation just based on doublets. So it's also a recommendation, I would say, that if your aim with your experiment is trajectory analysis, do not overload when you run 10x, because doublets are much more detrimental in that kind of analysis. So what can we do about detecting doublets? Of course, if we have a lot more detected genes, a lot more UMIs, if we have two cells in a droplet than just one, But that works really well if you're looking at only T cells, for instance, because then they should be about the same size, all of them, about the same amount of genes, and then clear outliers with high number of detected genes are most likely doublets. But if you have a heterogeneous tissue with a lot of different cells of different sizes, it will not work to just remove the high end of gene detection. So of course, after clustering, we can look at cells with gene signatures from multiple clusters. And then pretty much, I mean, if you see these streaks, if you also see that these have really high number of detected genes in here, it could be an indication that they're doublets. And of course, think back to what your doublet rate should be and think about if these proportions make sense. There are several tools. These are just a few of them that I've listed here that tries to simulate doublets. So basically, you take your data, you add together all the reads from two cells, randomly selecting pairs of cells. You put them into your data matrix, and you rerun PCA and so on. And then any cell that's most of its close neighbors are doublets, the simulated doublet, you would predict to be a doublet. And then basically all of these tools build on this with different ways of doing dimensionality reduction, calculating these scores and so on, but the idea is all the same. And this works quite well, I would say, to detect what I believe to be doublets. You never know, looking back at the data, either if it's correct or not. The issue is if you have very unbalanced cell populations. If you have one very large population and 3 small ones, that most of your simulated doublets will be these large populations, and it might mess things up a bit. But it's at least one way to make sense of what doublets you have in your data.
Is there some specific tool of these 3 that you recommend?
I've mainly just used these 2 first ones in my project. They give slightly different results, but fairly similar. So now I can't really say. Do you have any ideas, Paulo?
No, I mean, since they are quite similar as well, and I don't know, it's just testing, just like for most of the other tools that we'll show in the course. There are many replicates of the same thing over. So it's just testing, I think, and see what matches your data best.
Yeah, but then of course, I think it's also going back to knowing the biology of the system you're working with. Because I mean, in this case, I colored them by what I predicted to be doublet using, I think, doublet detection package. So all of these in cyan here are predicted doublets. This is a really overloaded sample, so we had quite a lot of doublets also. And then really looking into these, like signatures of these, especially like here we had 2 different cell types that we know we shouldn't have an intermediate of. So then I really trust it. On the other hand, along this path, we actually do have a differentiation pathway going on. So it might actually be the case that some of the predicted doublets here are falsely predicted. So it's really going back and forth with the data and thinking about what you have. It's also, I must stress, because I've read now several papers where people say, we removed all doublets, so there shouldn't be any doublets in there. I even saw a paper where they had some intermediate cluster that they listed as mix of cell types. But since they removed doublets, with one of these tools, it can't be doublets. And that is just completely wrong to me, because this is a prediction. They use the data they simulate. You can't assume that you removed all doublets. And you might actually remove some true non-doublets. It is just a prediction, so keep that in mind. There's no way to really remove all doublets for certain.
Can I ask a question?
Yes, of course.
So is that what you typically do to remove doublets, or would it be in a case where you want to keep them, just keep track of them?
It really depends on the research question at hand and what kind of questions they want to ask for it. Because depending on what you want to study in this data, the doublets might be more or less detrimental. As I said, if you want to study this developmental trajectory, for instance, then making sure that you don't have doublets in there is really important. If they still cluster really nicely into distinct clusters and you don't have these streaks here, then having a few doublets here and there will not shadow the signal of like when you do differential expression between clusters, and then it's not super important. It's also knowing, I mean, if in this case they loaded 10,000 cells, so I probably have a doublet rate of 10% or something. And if I get samples that are this highly loaded, I would always try to remove doublets. But if you only have like 1% doublets in your data, it might not affect your results as much. So you really need to think about how will doublets affect the conclusions I make from this data, and keep that in mind. It's also in some other projects, if I would, you can actually, what you can do, especially if you have specific clusters, if this part here ends up being one cluster, you can also look at differential expression of that cluster before you decide that this is really doublets and not an additional cell type, and then decide if you remove it. So there's not one straight answer, unfortunately. But I think it's good to run one of these, do the predictions, then you can decide if you want to remove them or not, or you keep them in, but still keep in mind that if you get a cluster with only predicted doublets, you need to be cautious about it.